leading to the rod cell's incredible sensitivity.
The following programme is for the second level science course, Biology, Form and Function. It looks at the maze-like systems of metabolic pathways, both in photosynthesis and in a metabolic disorder which can sometimes occur in milk production in dairy cows.
For this programme, we've come down to visit the National Institute for Research in Dairying at Chinfield. They run an experimental farm down here. One of their major aims is to maximise the quality of the milk and also the quantity of milk. In the programme today, we're going to look at two aspects of intermediary metabolism. A metabolic disorder that causes sometimes a reduction in milk production in cows, and we're going to look at the techniques utilised in the mapping out of the first metabolic pathway in plants, photosynthesis. Some of these techniques have now been utilised to map out intermediary metabolism in the production of milk. Scientists have been interested for years in trying to understand how plants biosynthesize their chemical compounds and how animals eating plants biosynthesize their compounds from the plants. Radioactive isotopes of lead were used to trace pathways in plants as early as 1913, but it wasn't until we had a stable isotope of carbon that we could actually trace the metabolic cycles inside cells. By bombarding nitrogen with neutrons from an atomic pile, nuclear physicists were able in 1940 to manufacture a stable radioactive isotope of carbon, carbon-14, which has a half-life of over 5,000 years. This really stimulated research because biochemists could use this powerful marker to label molecules and trace their routes through plants and animals. By the mid-40s, new ways had been developed to separate and isolate molecules of different sizes. The American biochemists Calvin and Bassam used these techniques to unravel the metabolic pathways of photosynthesis. Everybody knew that plants trapped light energy and produced sugars, but Calvin's team actually located the point in photosynthesis where the carbon from the atmosphere was incorporated into the plant. This was a breakthrough because photosynthesis is the process that generates the building blocks of plant and animal life. Calvin's experiment went through 4 stages. The first was the culture. Chlorella was the unicellular green algae used by Calvin in his experiment. These single cells were favoured over higher plant cells because they could be killed quickly with alcohol at different stages of photosynthesis, and the algae could be grown in vast quantity in laboratory cultures. Since Calvin's team realised that photosynthetic reactions occurred with great speed, they had to conduct the experiment with rapid and precise timing. Photosynthesis was started in a nutrient solution with ordinary unlabelled carbon dioxide bubbling through. For rapid equilibration, they switched to radioactive labelled C14 carbon dioxide, which was added in both gaseous and liquid form at the same time. Every 4 seconds, samples were drawn off into tubes containing boiling alcohol. This killed the cells and stopped photosynthesis almost immediately.
So Kelvin had samples from the photosynthetic process at various stages, and he could follow the synthesis of new radioactive compounds from stage to stage. In fact, even when the algae were only allowed to photosynthesize for 5 seconds, there were already several types of compounds showing radioactivity. At the time of Kelvin's experiment, a new technique called two-dimensional paper chromatography had been developed. This made possible the separation of small molecules. An extract of algae was spotted on a corner of filter paper, and a jet of warm air evaporated off the alcohol and water. A mixture of colored inks illustrates the process of separation more clearly. Time-lapse photography shows how gravity draws the solvent down the paper. The molecules separate out according to how fast they can travel through the paper. The paper is turned through 90 degrees, and the pigments are further separated by using a different solvent. To prevent evaporation, chromatographs were run in a water-saturated atmosphere in vapor-tight boxes and at a constant temperature. After several hours, the papers were turned through 90 degrees and run with a second solvent. Two compounds that travel at the same rate in one solvent may travel at different rates in another solvent. The chromatograph was placed in contact with X-ray film in order to locate the positions of the radioactive compounds. After several hours of exposure, the film was developed. The positions of the radioactive compounds showed up as black areas on the negatives. These pictures are called autoradiographs. They gave Kelvin a sequential display of the order in which photosynthetic compounds appeared. The compounds on the chromatograph paper were not visible to the naked eye, but they could be located by a device called a radiation meter. The small amount of radioactivity in the paper was not harmful. The autoradiograph was matched up with the appropriate chromatograph. This allowed the precise location of the radioactive compounds. The final step was to ring them and cut them out. Once the different compounds were isolated on individual pieces of paper, they could be eluted and collected in vials, ready for analysis. Identification by standard chemical techniques took a lot of time in those days, but fortunately, chemists could usually predict the type of compounds present. They would therefore mix the unknown radioactive sample with a known chemical. This mixture was then spotted onto chromatography paper and run with various solvents. By making an autoradiograph, the unknown radioactive compound could now be located. A special reagent spray made the known non-radioactive compounds visible. Then, by comparing the positions and shapes of the two compounds on the film and on the paper, it was possible to tell whether or not they were the same chemical.
Well, that's the technique. But what did the experiment actually show? They were able to localize the radioactivity during the sequence of events of the photosynthetic pathway. If they let the experiment run for 2 seconds, 70% of the radioactivity was localized in a 3-carbon compound called phosphoglyceric acid. Let's call it PGA. Shortly after that, the radioactivity was also found in 2 3-carbon compounds, phosphoglyceraldehyde and a ketone isomer of this. And then, shortly after that, the radioactivity was found in another compound, a 6-carbon compound, a hexose sugar. If they let the experiment go for more than 30 seconds, they found that there was an increase in radioactivity in some of these particular compounds. The only way that could happen is if the process was cyclical and more than one radioactive C14 carbon was in one of these individual compounds. The real point of the experiment, though, was that they were able to isolate the compound that reacted with the carbon dioxide of the atmosphere.
For a long time, Kelvin had looked for a 2-carbon precursor to combine with carbon dioxide to make a triose compound. In fact, it was a 5-carbon compound called ribulose bisphosphate, which reacted with carbon dioxide to yield 2 3-carbon compounds, 2 molecules of PGA. The enzyme catalyst in this reaction turned out to be ribulose bisphosphate carboxylase, probably the most abundant enzyme in the world. Labeling the molecules with radioactive isotopes also revealed something else. By breaking the molecules down into individual carbon atoms, they were able to pinpoint the position of the carbon-14 in the structure of each molecule. This gave them a clue to the type of reaction that had taken place. For instance, this hexose sugar has activity here at carbon atoms 3 and 4, suggesting a condensation reaction between one molecule of the aldehyde and one of the ketone. This in turn suggested the sort of enzyme involved, in this case, an aldolase. Actually, Kelvin's team found radioactive carbon in many other compounds, and this linear route is only a fraction of the many interconnecting pathways that make up intermediary metabolism in animals and plants.
We humans haven't got the necessary enzymes to efficiently convert green plants into energy. But we can use biological converters like these cows to do it for us and to give us the dairy products. Now that milk tastes fine, but that milk smells awful. It smells like nail polish remover. In actual fact, the milk has got acetone in it. That tells the farmer that there's something wrong with the cow. It has a metabolic upset. It's called ketosis, and acetone spills over into the milk. The ironic thing about ketosis is that it happens in cows that produce a very high milk yield. To understand why this happens, we really have to look at the lifestyle of a modern dairy cow on a dairy farm.
Dairy farming is big business in this country, and to be competitive, farmers have to be efficient. EEC regulations control the quality of the milk, and the farmers want to maximize the quantity of output. Domestic cows have come a long way from their ancestors in the wild, who produced milk only to suckle their young. As we demand more output from the cow, so we have to ensure the right input, the correct diet. A cow is a ruminant. She eats a large amount of forage crops like grass and digests it in her stomach, an enormous four-chambered vat full of microorganisms that break down polysaccharides like cellulose and ferment them into smaller molecules. From this broth, the cow absorbs amino acids, fatty acids, and glucose into the bloodstream. These raw materials have several possible fates. They can be burned as fuel to generate energy. They can go to build up protein as muscle.
They can be stored as energy in fat or fed to the young as milk. The record production of milk per day in a cow was about 85 litres, the height of this stack of bottles. To get that much milk, the cow's udder had to utilise 4.8 kilograms of glucose, 3 kilograms of amino acids, and 3 kilograms of fatty acids. To produce that much milk, something like 40,000 litres of blood had to pass through the udder. And the udder cells were synthesizing molecules at the rate of 3 times 10 to the 19 molecules per second. In the everyday world, most cows produce about 40 litres of milk a day. And even to produce that, they have to have the right amount of protein, of fats, and amino acids. But one of the critical supplies to the cow is the amount of glucose. Reduce the amount of glucose, and you reduce the amount of milk production. It's been shown by using radioactive glucose that practically all the glucose goes from the blood into production of lactose, the main disaccharide in milk. You've seen how hexose sugars are handled in the carbon reduction cycle in plants. Now let's have a look how they're handled in the metabolism of the cow.
Well, it's relatively simple to make lactose, a disaccharide. 1 molecule of glucose, which is just a cyclized hexose sugar, and 1 molecule of galactose, combine to make 1 molecule of lactose. But although the cow absorbs some glucose from grass in the rumen, most of the glucose and galactose that go to make lactose are biosynthesized from starting materials, which are absorbed as fatty acids or amino acids. The route to glucose production, called gluconeogenesis, then starts at the tricarboxylic acid cycle, the main energy-generating system. Fatty acids are broken down ultimately to acetyl coenzyme A, a 2-carbon compound. This molecule effectively drives the TCA or tricarboxylic acid cycle. 1 molecule of acetyl coenzyme A combines with 1 molecule of oxaloacetate to make a molecule of citrate, a 6-carbon compound. The cycle involves the loss of 2 carbon atoms as carbon dioxide to return 1 molecule of oxaloacetate to the beginning of the cycle, ready to combine with a new molecule of acetyl coenzyme A. 1 turn of the cycle generates many molecules of ATP. Amino acids can join this catabolic process at various places. Some are broken down to a 3-carbon compound, pyruvate, and others enter directly into the TCA cycle. When glucose is being used as an energy source through glycolysis, it finally enters the TCA cycle as acetyl coenzyme A. The opposite process of gluconeogenesis, the anabolic process of building up, has a slightly different path. It starts with the conversion of oxaloacetate to phosphoenol pyruvate, a step that avoids any intermediate form such as pyruvate or acetyl coenzyme A. In fact, These steps are essentially one way, and the enzymes that catalyze these transitions characterize the two processes. After that, all the steps to and from lactose are reversible.
Fluid models are useful ways of describing some aspects of intermediary metabolism inside cells. Look at this model, for example. Imagine it inside the cells of the udder producing milk. These tubes here are enzyme-directed pathways, and here down there are two taps, which are the controlling aspects for these pathways. Substances like fatty acids and amino acids that get absorbed from the alimentary tract of the cow are converted into acetyl coenzyme A, as is building up in this reservoir here. Acetyl coenzyme A comes down here and condenses and links with oxaloacetic acid from the TCA, or tricarboxylic acid cycle. They pass round the cycle, generating new oxaloacetic acid to fuse with the acetyl-CoA, and also producing ATP, or energy, for the cow's usage. But also from this cycle, the cow is pulling oxaloacetic acid from this pathway to go through the biosynthesis into glucose and galactose, and ultimately to lactose, the disaccharide of milk. Under normal circumstances and normal cows, everything's in the steady state. The levels here and here all balance out and everything works fine. But in those high-yielding cows, the ones who produce very large amounts of milk, a large amount of oxaloacetate is taken out for milk production, and the level of oxaloacetate here starts to drop down. This means that when this level is being pulled out here, there's not enough oxaloacetate now to link up with the acetyl coenzyme A. And the acetyl coenzyme A in this particular reservoir starts building up and up and up. Because not enough energy is produced in the cycle around here, because it's not turning around very efficiently now, the cow needs energy and starts to break down fat into fatty acids, which produce even more acetyl coenzyme A, so this builds up even more so. Well, normally the cow can handle a certain amount of acetyl coenzyme A going through to ketone bodies, which in fact some tissues need. But this vast excess of acetyl coenzyme A produces far too much ketone bodies. It spills out of the cell into the bloodstream and then finally into the milk. In this condition, the cow is in ketosis. The cow loses its appetite, it doesn't eat, and the milk production falls off. How does the farmer get over this problem? The farmer has to ensure that each cow gets the right diet, a diet providing enough glucose rather than fatty acids or amino acids. The problem comes about because over the years we have selected for such high milk yields that we can no longer rely on the cow's normal metabolic control to regulate the pathways leading to milk production. When everything's fine, the cow's happy, the farmer's happy, and you get your daily pint.